Good morning, everyone. I am welcome you all to this 12th day of CSIR Life Science question and answer discussion session. In this session, we are going to discuss about the Unit 4, okay, Part C questions in the Unit 4. Okay, as per the our schedule, we are finished Part A discussion and Part B discussion. Okay, past 11 days, we are totally covered the of the Part A and Part B questions. Hereafter, we are going to spend the time for the part C, that is the major part in a CSIR examination. In this part C, we have another more 19 days. Okay, it's a 30 days program. In this upcoming 19 days, sorry, this days we will going to discuss about all the part C questions. Why I am focusing this part C means if we are cover this part C portion, that will expected in a part B also. So this part A is fully going to discuss the concept relevant and the research relevant informations. And one more important thing about the part C. Here, part C question, there are three important units, that is the fourth unit, fifth unit, and 13th unit. What are all the units? Fourth unit is a cell communal pathway. It's a very important topic. Past 10-year analysis. This unit have a 7 part C questions. If you are attend one part C question, you can get 4 mark. So if you are thorough in a fourth unit, you can answer maximum 7 question. So if you are attending this, if you are well versed in this part, unit 4 means you can easily achieve 28 marks in the part C. And part B also there are 3 questions in a fourth unit. So if you are fully thorough in this fourth unit, you can achieve 28 plus 6, nearly 34 mark. Okay, this is the major thing about this unit 4. Likewise, if you are well versed in the unit 4, 5, that is develop, and 13th unit, that is methods in biology. So 34 plus 34 plus 34, you can easily achieve the 1 or 2 mark if you are thorough and well versed in the topic of the cell communication and cell signaling. And another topic is a developmental biology, unit 5, and unit 13, methods in biology. If you are well versed in these 3 topics, you can easily achieve 1 or 2 marks. So as per our focus, this part C discussion will give you the brief knowledge about the unit, this particular units. The upcoming 19 days we having the including today, we are having 19 days. This upcoming 19 days, first 9 days, okay, first 9 days we are going to fully discuss about the unit 4, 5, and 13. First 3 days for today, tomorrow, and day after tomorrow. It's all about the fourth unit, part C question discussion. And upcoming days, the first 9 days of our session is going to be on about the part C discussion of this important units 4, 5, and 13. After that, finishing this important topic, and then 10 more days we will schedule the upcoming content. Okay, here we can start this session. Before entering the session, please let me know, am I audible? Can you see my slides? Is it visible? Please let me know through the chat box. Is it audible? Am I audible? Is it visible to you? Discussion will give you. Yeah, okay. Thank you for your response, Rasika. We will start the session. About the in unit 4. First of all, whenever you are facing the part C, that must be in a large paragraph. That we can't expect a one line question. There is a big paragraph like research knowledge. First, you want to read thoroughly about the question. Okay, first we need to read the question. Okay, I think my slides are visible today. I'm changing the live slide design. Mouse bone marrow cells were fractionated to derive stem cell antigen plus minus 1 positive SCA minus 1 positive cells. Bone marrow cells were fractionated to derive stem cells antigen. This antigen named minus 1 positive SCA minus 1 positive cells. These cells were cultured with interleukin 3. Here, this is the keyword. Here, the cells were cultured with the interleukin 3. Are granulocytes macrophage are granulocyte macrophage colony stimulating factor or macrophage colony stimulating factor. Here first one is granulocyte macrophage colony stimulating factor or macrophage colony stimulating factor or granulocyte colony stimulating factor. Granulocyte colony stimulating factor. There are three compounds, four compounds are added. One is interleukin 3 or granulocyte macrophage colony stimulation factor or macrophage colony stimulating factor or granulocyte colony stimulating factor. Most numerous and varied colonies were obtained in the culture stimulated with, where we can get the most numerous and varied colonies. This kind of process happened with the culture stimulated with the interleukin. The interleukin linked interleukin added culture shown the now numerous colonies and numerous number and varied colonies. This is the they are if okay you can ask how can we where can we study this kind of questions are fully related to the research. Okay, whenever you are studying the topic we want to do the research journal based study. So that is too complicated for the BTech and basic beginners. Beginners can maybe struggle with this kind of question because in a theoretical we are well versed in the theory because theoretical that is the part B portion. But many of the questions in part C it's fully on the research oriented. Please try to do the research article studying in part in the preparation. Meanwhile, the preparation. So here the once again I'm read the question. Mouse bone marrow cells were fractionated to derive stem cells antigen. For that, in this culturing process, they added the different factors, that is interleukin 3, granulocyte macrophage colony stimulating factor, macrophage colony stimulating factor, granulocyte colony stimulating factor. The most numerous and varied colonies were obtained in the culture stimulated with interleukin 3. Next question. Cancer causing gene can be functionally classified into mainly three types. That three types are gene that cellular proliferation, tumor suppressor gene, genes that regulate the apoptosis pathway. These are all the three main types. Epstein-Barr virus. Here, Epstein-Barr virus that causes cancer by modulating apoptosis pathway. The keyword they are this virus modulating the apoptotic pathway contains a gene having sequence homology with which of the following gene? Okay, here the keyword is apoptotic pathway. For this question, we want to know what are all the genes are involved in the apoptosis pathway. Here, if we are know the genes that playing a major role in apoptosis pathway, you can easily answer. Containing this gene, that virus, Epstein-Barr virus that causes cancer by modulating apoptosis pathway, contains a gene having sequence homology with which of the gene? Okay, here the answer is the BCL, mention the BCL2. In the apoptotic pathway, BCL2 is the initial triggering molecule. If we are modulating the gene, this gene modulating the apoptotic pathway, this viral affecting this BCL2, that BCL2 alteration makes a change in the apoptotic pathway. And okay, the caspase 3 is also the important. But in this question, they asked this virus modulating pathway. If that caspase 3 is a final change, okay, final change of the molecule, in that step we can't modulate the pathway. We get the modulating the pathways because of the BCL2 gene. Okay, next question about the this unit 4 part C, toll-like receptor 4, TLR4. It's a famous and well studied receptor. Toll-like receptor with responsiveness of lipopolysaccharide, responsiveness to lipopolysaccharide, LPS, and endotoxin that causes lethal endotoxic shock. The mice deficient in a toll-like receptor, C mice, were injected with Escherichia coli. In additional, some BALB-B mice were injected with some bacteria alone or with the interleukin 10 antibody to the lethal effect of bacteria were. Okay, here the statement is the provided the statement first given information. The toll-like receptor is associated with the responsiveness of to the lipopolysaccharide and endotoxins that are causes the lethal endotoxin shock. This is the and they are provided the question. That question having the data, the mice deficient of TLR4, that is the toll-like receptor 4. So deficient means there is a lagging of TLR4 and BABL and okay, the mice are don't having the they're deficient in the toll-like receptor. That two type of mice are there, BALB-C mice, and one other thing is BALB mice, bar B minus. It's a C minus, a C mice, and this is the B mice. C mice is injected, the B mice is injected with the same bacteria. Okay, this bacteria Escherichia coli alone, or it may injected with the interleukin 10. The mice resistance to the lethal effect. Okay, this lethal effect is resisted by the mice, so there is a no endotoxin shock. The effect to the bacteria, where which mice is make a resistance. If the mice is injected with VALB, bar B mice receives the bacteria and that having the anti interleukin 10. So this kind of injections makes the mice to resistance for particular effect of bacteria. Okay, this kind of the antibody assisted injection of the bacteria. Here they are antibody, they are anti-interleukin 10 assisted injection of bacteria that makes the resistance to lethal effect. This is the concept is whenever the mice is injected with the antibody that makes a resistance to lethal effect. The next question is glucose is mobilized in a here the next question is glucose is mobilized in a muscle when epinephrine actives GS alpha in an experiment in which muscle cells were stimulated with epinephrine. Glucose metabolization was observed even after the withdrawal of epinephrine. Could be this is the reason they are asking. The reason could be due to the presence of CAMP, cyclic AMP phosphodiester inhibitors, the esterase inhibitors. So they provided the statement, ask the reason. In this way we can see, okay, what is the reason, what is the respective reason for the respective question. As per the question, glucose is mobilized in the muscles when epinephrine is activates the alpha, GS alpha. This is a statement. In an experiment, which muscle cells were stimulated with the epinephrine. So there was the epinephrine in the muscle cell. Glucose mobilization was observed after the withdrawal of epinephrine. After withdrawal means that is excreted from the muscle cells. So this is due to the presence of CAMP phosphodiester inhibitors. Intracellular pathogen like Mycobacteria, Salmonella, Leishmania and Listeria survive in macrophage by modulating host cellular machinery. Okay, they are modulating host cellular machinery. In order to study the fate of these intracellular pathogens in macrophage, cells were labeled with lysotrax red. This is the labeling molecule labeled with lysotrax red and infected with GFP labeled organism. After the 2 hours at 37 degrees Celsius, cells were fixed, stained with anti-transferrin. Okay, stained with they are labeled with lysotrax red and stained with anti-transferrin receptor antibody and probed with the secondary antibody conjugated blue dye. Okay, first is a labeling molecule, then staining, after that staining antibody, this is a primary antibody, then adding the secondary antibody that is conjugated with the blue dye. Cells were viewed under the confocal microscopy. This is the statement, the whole statement. There are microorganisms, that microorganisms are intracellular pathogen. It's modulating the host cellular mechanism. This pathogens in a macrophages were studied in a laboratory scale that are labeled with lysotrackers and infected with the GFP labeled organism. And after the 2 hours, that are stained with anti-transferrin receptor and probes with the secondary antibody. This anti-transferrin receptor is the primary antibody, then adding the secondary antibody with the dye, blue dye, and the provided it's we are they are visualized under the microscope. What is the observation? This observation, the GFP is labeled GFP, labeled with mycobacteria, salmonella, and listeria were located in a same compartment labeled with blue dye. Whereas GFP Leishmania colorized with a red label compartment. Which of the following statement is a true observation? Okay, there are four organisms are there. In the four organisms, three organisms are labeled in the same compartment with the blue dye. Leishmania alone is labeled with the red color compartment. What is the reason behind it? In this statement, which from the statement which is the following is, as per the question and observation, the Leishmania reside in a lysosome-like compartment. Okay, it's having the lysosome-like compartment. So only it's labeled with the red color. Here they mention the lysotracker lid. Okay, here they are labeled with lysotracker means that red is passed through the lysosome. That is the lysotracker red. It can be passed through the lysosome. If the cell absorbing the red compound, red GFP, that allowing the compound into the lysosome. So only they mention the reside in a lysosome like compartment. So this study, whenever you are reading the question, just imagine the real life situation. Then only we can easily answer the question. The next question, macrophages were collected from BALP mice, C mice. Okay, the one statement, it's collected from the BAE mice, CD40 deficient mice, and CD86 deficient mice, and ICAM1 deficient mice. They are all collected from various cells. These macrophages were co-cultured. Co-cultured means it's a the mixture cultured with the, when additionally added with the LCME peptide specific T cells. CME peptides for 3 days. These cells were recovered and recultured with the BALB C derived macrophages in the presence of the peptides. First, the cells are all the macrophage cells cultured is kind of mice. After that, that is co-cultured along with the LCME peptide specific T cells. This for 3 days. This after this, the cells were recovered. The cells were recovered and co-cultured with the BALBC derived macro. During the last 12 hours of co-culture, 3-H-thymidine was added to the culture. This is the labeling. It's a thymidine. It's cultured with that cell. The cells were harvested and 3-H-thymidine is. The highest incorporation was observed in a BALB bar C macrophages T cell and co-culture. So after this step, okay, this co-culturing step, macrophages were this step, okay, after the cells were recovered and co-cultured with the BA cells in presence of the peptide. After this step, they are observing this cells with H-thymidine. This happened with the BALBC macrophage cells and cells co-cultured. This is the reason. This is a fully research based question. It's an advanced research question. they can't able to understand in a theoretical manner. So just imagine the culturing means that is the animal cell culturing technique. If you not follow up or if you not pop up the question, please tell me, because actually biotechnology, microbiology and biotechnology, microbiology and biochemistry peoples may know about this concept, the cell culturing. But if you not understand, please let me know. I will make the changes in the study mode. Okay, next concept is the patient undergoes liver transplantation, and during the course of post-operative treatment become a success to injection. The patients can treated in a two different correct. The correct statement is treatment with antibodies reducing the infection but retaining the transplant. The treatment with antibodies reduce the infection but that retaining the transplant. Upon ligand binding, the cell surface receptors move laterally to capped and internalized. There, this is the statement. Upon the ligand binding, that cell surface receptors move laterally to be capped and internalized. Leishmania, a protozoan parasite, can cause several receptor on macrophages to get internalized that want to be entered, so that makes a more several receptors. One of them is toll like receptor that binds lipopolycon on leishmania. On internalized Once internalized, the parasite is destroyed in the phagolysosome. Which of the following treatment of leishmania infected macrophages will result in lowest parasite number in a macrophage? Okay, this first two lines are statement. Oh, sorry, first three lines are statement. Just read out again. Upon ligand binding, cell surface receptors move laterally to be capped and internalized. Leishmania, a protozoan parasite, can use several receptors on macrophages to get internalized. One of them is a toll-like receptor that's helpful for the penetration internalization. Binds lipophospholipoglycan on leishmania. Once it's internalized, the parasite is destroyed in a phagolysosome. Which of the following state treatment of leishmania infected macrophage will result in a lowest parasite number in macrophage? So the reason behind it is both beta MCD and ammonium chloride. They this macrophages are treated with the beta MCD, and the ammonia chloride means that this kind of treatment help helpful to lower the number of parasites in a macrophage. Okay, the next question about on the structural region of a nuclear receptor shown in a diagram. The following predictions were made. Here the question statement is based on the structural region of a nuclear receptor in the shown in here the structural regions A, B, C, D, E, F. These are all the structural regions. This is the protein like structure. This having amino acid N and carboxylic N. So they are asked the question, finally one question, and provided the statement. Region F is responsible for binding to the ligand and containing two zinc finger like binding motif. Is it right or wrong? We want to check. Then here they are zinc finger like binding motif. How can we tell this zinc finger like this is not present and the so the B is eliminated. Receptor with AB domain generally associated with the chaperones do not bind to DNA. Yeah, right. DNA, the amino acid is in it's having they are having the A and B receptors that not able to bind with the uh, that binds with that compound. Region E indicates that receptor associated with chaperones which protect the nuclear hormone receptor. That this in this statement, our statement, it's having the nuclear hormone receptor. This is the it protects the nuclear hormone receptor. It's not acceptable one because this kind of chaperones are not relevant to the nuclear hormones. And so R is wrong one. Yes, the region C containing P box and T box required the dimerization of the direct with the phosphodiester bond. So intermediate region it makes a phosphodiester bond. So it's acceptable. Yes. The so answer is the Q and yes. Next part is monoclonal antibodies can be potentially used as a therapeutic target. This is a statement. A major advantage they can especially target a abundant cell. However, there is a practical difficulty. Monoclonals are raised in a mouse and therefore is expected that an immune reaction will develop if there is an injection into the human. It is therefore necessary to humanize the monoclonal antibody by taking a human IgG and replacing the CDRS by those derived from the mouse monoclonal antibody. They are replaced with uh, this replacing this human IgG. Okay, they are it's a taken one. Okay. Next, a fixed smear of bacterial culture is subjected to the following solution in the order listed below. Appear the red and appear the red. So smear we want to make, that is the first one is the carbofucosins, that is the heated condition, acid alcohol and methylene blue. The bacterial culture is subjected to the following solution. Okay, we all know the staining technique, the gram staining. Different staining methods are there. Here the methylene blue staining is played here. Carbol fusins and acid alcohol and methylene blue. The bacteria stained by the method identified as this method is helpful to identifying the acid fast mycobacterium species. The acid fast microbacterium species are characterized by this kind of smearing technique. The smearing technique is having the carbol, the fuchsins, and acid alcohol and methylene blue. Next question is the survival of intracellular pathogen depend on the level of pro-inflammatory and anti-inflammatory cytokines in a macrophage. In an experimental condition, mycobacterium infected macrophages were treated with interleukin and interleukin 2 or interleukin 12 for 4 hours and 37 degrees Celsius. Untreated cells were used as a control. Cells were lysed and number of bacteria in each set was counted by measuring colonies forming unit. Which of the observation is true? Paragraph. So in this paragraph, they are providing the survival of intracellular pathogen depends on the uh, pro-inflammatory and anti-inflammatory cytokines macrophages. This is the statement. In an experimental condition, in the research, the mycobacterium is infected the macrophages were treated with the interleukin 6 and 12 or 12 for 4 hours. This is the this microbacterium is a infectious microbe which treated with the cells. After that, that cells are treated with the IL interleukin 6 and interleukin 12. Untreated, okay, without the interleukins, the untreated cells were used as a control, okay, right? Cells were lysed and number of bacteria in each experiment sets were counted by measuring colony forming unit, okay. Here the output is measured as a colony forming unit. Which of is true? Interleukin 6 is treated with the contain the 6, the C is the answer. The interleukin 6 treated cells containing more intracellular bacteria than the control. Because why? Because means here you can see the first statement, survival of intracellular pathogen is depends on the anti-inflammatory cytokines. These are all the cytokines. So if that cells are added with these cytokines that are utilizing the cytokines and it's grown in a more number, the interleukin 6 contains more intracellular bacteria than the control. The next question is, many of the cancer carries the mutant p53 gene, while some cancers have normal p53 gene. P53 activates P21, that is a VAF1, which inhibits G1 bar S cytokines, that CDKs, phosphorylation of retinoblastoma protein. This is the two character. So one is activation of P21 VAF1, it's inhibiting the G1S phase, and phosphorylation of with normal P53 gene good. It's here the question is, this answer is C. D, it's express the inactive form of G1S cytokines. The next, now impulse or action potential is changes in the permeability of axon membrane to Na plus and K plus ions. The depolarization of membrane beyond the threshold level, it leads to Na plus following to the cells, and the changes in the membrane potential to the positive value. The K plus allowing the K plus to follow outwards, ultimately restoring the membrane potential to restrict the value. The number of that Na plus and K plus channels operate in the opposite direction, because why it's acting the opposite direction. So if there is a difference in the Na plus and K plus concentration on either side of the membrane. Next question is, bacterial response regulator turns on gene A in its phosphorylated form. The amount of A shows a sharp and steep rise at the threshold concentration of a signal sensed by cognate sensor. This is most likely due to the answer C, cooperativity in a binding of response regulator to the target gene A. Here they has provided the statement, the bacterial response regulators turn on a gene A. So its regulators turns on the gene A. This amount of A shows the sharp and steep rise in the threshold concentration of the signal sensed by the cognate sensor. So its amount is gene A become high. The most likely due to the answer C, cooperativity or in a binding of the response regulator to the target gene. So if the bacteria it's attached on the receptor, the regulator that makes the turn on the gene A, so that is the higher number of genes. So this regulator is target, this regulator it's correctly binding to the target gene A. Next question, you are given a group of 4 mice. Each mouses are immunized with the keyhole limpet hemocyanin or asobenzine, arsenate or lipopolysaccharides or dextrons. 4 weeks later, sera were collected from these mice and antigen-specific IgG1 and IgG2A ELISAs were performed. Only one of the mice is shown positive response. Why it's only one mice shown a positive response? Why? Because the whole lymphat hemocyanin primed mouse alone, it's shown the positive response. Why? Because this they are making this um, they are expressing the gene. Okay, next is very interesting question. T cell proliferation in vivo is to be analyzed. The cells are labeled with the CFSE, that is the fluorescent probe CSFE, and injected CD86. Deficient mice, and it's injected with the BALB mice or BAC mice along with the required antigen. 3 days later, the cells were recovered and analyzed by the flow cytometer, because this is injected with the fluorescent probe, so that is analyzed by the flow cytometer. Which of the following is logically correct? So here the BALB bar C, it's shown in the lines. CD86 deficiency is the daughter lines. Here the fluorescence is high, it's improving. Here this is the event. As per their question, they provided the correct situation is B. How it is B means the CD86 deficient is no, it's normally increasing, and it reach at one particular point. But the BALB, that sorry, it's CD86 deficiency is increased, but the BALB C is it's a smooth improvement is there. Why? Because the cells are first injected, they are labeled with fluorescence and injected with this and this injected in this kind of mice, so that mice are specifically having some antibodies in their own body. So this antigen, the T cells are they injected means that makes some uh, alternative changes in their immune system. So in this immune system response are leaded with the fluorescence molecule. So this B answer is the right one. After the 3 days, that it's a event. It's a CD BALB bar C injected cells where that mice shown a smooth response and that increased response, and CD86 deficient mice shown some alterations. Then intracellular transport and cytoskeletal organization of the cells is regulatory exchange of different small molecular weight GTPase of RAS superfamily. Overexpression of which of the following GTPase modulates an actin cytoskeleton of HeLa cells? This is a well studied experimental research work, because this HeLa cell and ATP cell lines are expressing that GTPase signaling and RAS signaling pathways. Here the correct answer is a rho in a GTP bound form. Okay, rho, that is a protein which in a GTP binding form. The cells were isolated from a breast cancer patients. That cells were injected with the nude mice. There is no cancer. They were derived into 4 groups. Group 1 receives EGF receptor conjugated with methotrexate. Group 2 receives the transferrin receptor. Group 3 is injected with mannose receptor conjugation with methotrexate. And group 4 is received the same amount of free drugs. There is a no drug, that is a free drug, but some of this amount of free drugs alone they are injected. If in which of the following case is a tumorogenic, index would be minimum. the tumorogenic index minimum in the EGF receptor conjugated drug. So that is helpful to treat the their expression. Okay, next question is this is a biggest question but easy to answer. In a tissue, cells are bound together by physical attachment between cells to cell or between cells to extracellular matrix, following the same characteristics, some characteristics junctions. These are all the factors. Here, which they are asking which of the following combination is an eliminate the not correct. Is a combination is not correct. Another one thing, statements all are correct. The combination of statement is alone not correct. So adhesion junctions are cell-cell anchoring junction connecting actin filament in a one cell with that in the but it's not correct when they are connected with another statement. Desmosome. Okay, desmosomes are cell matrix anchoring junction connecting actin filaments in one cell to extracellular matrix. This is it's another statement. Cap junctions are channel forming junctions following passage of sample water soluble molecule from cell to cell. It's right. Are accumulating junctions which seal gap between two cells. Hemodesmosomes are cell matrix anchoring junctions connecting the intermediated filament in one cell to extra statements. These statements combinedly, some of the statements are wrong. Like here 1 and 2 is comparing this two states while comparing two statements, that is the combination is a not correct one. Okay, this is some of the important questions about the unit 4. Part C questions. Tomorrow we will discuss the remaining part of the unit 4. Okay, first 3 days, 12, day 12, 13, 14. It's a time to study the part C questions of the unit 4. If you have any questions, please post it in a chat box. Okay. 56th questions. I think this question, Rasika, is this question 60, you are asking 66th question. Sorry, 56th question. I don't know which 56th. Okay, fine. Tomorrow onwards, I will include the slide number. That is better to understand. Sure, I will send the study material for today class, for today questions. Okay, actually, the 256 questions are there. I don't know which you are asking which question. Yeah, okay, Rasika, I will tell this. Okay, here the question is you are given a group of four mice. You're having the 4 different mice. Each mouse are immunized with keyhole limpet hemocyclidine. One is injected with this molecule hemocyanin and asobenzene arsenate. It's another mice injected with this. Another mice injected with lipopolysaccharide. Another mice injected with dextrons. And this in this case, the after 4 weeks, the seras are collected from these mice. Each mice are collected the antigen specific IgG1, IgG2A ELISA were performed. Okay, in that ELISA experiment, we are adding the antibodies to the serum. So, if the here they mention antigen specific IgG1. So, this IgG1 is specifically bind with the antigen IgG. If in this experiment the only one thing alone getting positive response, the mice only the one mice shown positive response. So the mice is injected with the that asobenzene, arsenate, lipopolysaccharide and the dextrons are not shown any immune responses, that are not that our body is accepting the chemicals. But the mice injected with keyhole hemocyanin. This is the one compound which makes the immune response. Okay, in this here the molecule is a complicated large protein like structure, this having multiple subunits. It's a metalloprotein. This no in this if this is a complicated structure, so our body recognizes this as a foreign particle, and that makes a immune response against that particular compound. So in that mice, that immune response is stimulated against this chemical the lymphat hemocyanin. This chemical producing the antigen IgG, and it's producing the IgG2 in their mice. So this serum having the IgG antigen and IgG2 antigen. Whenever you are doing the ELISA, with that ELISA, the serum is added with the IgG antibody. IgG1 is antibody. Okay, this producing the IgG antibody, G1 antibody and the IgG2 alpha antibody. So there is a binding happen between the that antibody and this antigen, that make a positive response. The result, the color change occur in the ELISA experiment. This is the reason for only this mice shown the effect of this. I think you can understand now. The other compounds are our normal human proteins, these are all the simple structures. Okay, this knot is recognized as a foreign particle. This keyhole lymphat hemocyanin alone is recognized as a foreign particle. This is the complex metalloprotein. So this protein is recognized by the immune system. This immune system is considered, okay, this is foreign particle, we want to produce the immune response. This is very strong and well known immune stimulant. I think you got it. If you have any queries, please let me know in Telegram group. Okay, fine. Thank you so much. Okay, thank you for your response, participants. I will welcome to tomorrow's session. Tomorrow we will discuss the remaining questions of Unit 4, Part C. Okay, we will discuss more in tomorrow class. Thank you, everyone.